Hello, everyone. My name is Jessi Kreder, and I'm currently an undergrad student earning my biology degree at Lexington University. My main focus is on botany and environmental research, and today I'll be talking about microplastic contamination within agricultural, wetland, and floodplain areas. So I conducted this research over the summer with my mentor, Dr. Zimmerman, and I hope to shed some light on the importance and severity of microplastic pollution in terrestrial areas. So, what are microplastics and why should we research them? Microplastics are small pieces of plastic, usually defined as pieces less than 1 to 5 millimeters. And microplastics can be placed into 3 different categories: fibers, fragments, and beads. Fibers are extremely thin and long, should be a consistent thickness, not tapered at the ends, and should be clear or one solid color. Whereas fragments can be irregular in shape with sharp or jagged edges, and beads are round colored or clear pieces. So the 3 photos below are pictures of microplastics found within the soil samples I studied. The first and second pictures are good examples of what microplastic fibers within soil look like. And the third photo, it can be a little bit difficult to see the full microplastic, but the red plastic particle under the soil is an example of what a fragment might look like. However, the question is, why should we study them? Plastic production and consumption have become a very common aspect of everyday life for many countries around the world. Since single-use plastics replace traditional materials like glass and aluminum, this has occurred because they have quick and cheap manufacturing, their versatility, and they're considered much more sanitary. The production of plastic has increased each year and reached 35.7 million tons in the United States in 2018, with only about 3,000 tons of the plastic being recycled, according to the EPA. So with this increased use in plastic, the environmental consequences have become a very prevalent issue in the environmental studies, such as the usage of non-renewable resources for manufacturing, the long lifespan of plastics littered all over the world, and the influx of microplastics in essential water sources. With microplastic pollution on the rise, there are many concerns surrounding the effects these small particles might have on the environment and the human body. In recent years, microplastic contamination in aquatic environments has been studied a great amount in order to assess their consequences. However, microplastic contamination within terrestrial areas is still a very new concept and has very little research and data regarding the issue. This lack of information could be due to the difficulty of microplastic extraction from the soil. It could be due to its complex soil matrix and maybe the varying microplastic accumulation throughout dry lands. As you can see in the photo on the right, this is an aquatic area that has been polluted with microplastic. It's relatively easy to remove these particles by using a variety of filtration systems. However, for extracting microplastics from soil, it can be difficult to discern between a soil particle or a plastic particle, and separating the soil and plastic effectively is a little more in-depth. Since plastic production occurs on land, it's believed that microplastics, before they enter the ocean and other water sources, they penetrate terrestrial ecosystems and lead to environmental issues. So due to the future environmental risk of microplastic pollution on terrestrial environments, it's important to expand our data and our knowledge regarding this issue in order to fully understand their impact and help us restore our environments. Furthermore, the data surrounding microplastic contamination within agriculture areas, wetlands, and floodplains is very scarce. So by evaluating the amount of microplastic pollution in an agriculture area, a better conclusion can be made about the effects microplastics have on the environment. So during this experiment, I used density separation to extract microplastics from agriculture wetlands and floodplains, and the found particles were quantified. So due to the knowledge of the presence of microplastics in agriculture and wetland areas, I predict that microplastic contamination among the wetlands would be evenly distributed. Also, I expected that the floodplain areas would have a higher concentration of microplastic pollution since it receives runoff from the croplands and the wetlands and it also has a connection with a nearby river. And finally, it was predicted that the smaller microplastic particles would be higher in concentration due to their small size and how easily they can move through the soil. For this experiment, I collected sediment samples from the Franklin Research and Demonstration Farm in Lexington, Illinois. There are 3 plots of land that drain into 3 separate wetlands, the gully, the east wetland, and the west wetland as seen in the photo. So rainfall and groundwater accumulate in the soil and then enter the tile system, draining into the separate wetlands. Each wetland, when flooded, drains into one large floodplain, and the floodplain also receives overflow from a nearby river. So these wetlands in the floodplain were selected because each piece of the land caught the runoff from croplands and the possible pollutants and plastics found in the rain and soil of the crops. And they also contain an abundance of wildlife that could affect the amount of plastic pollution. Additionally, this area was chosen to help quantify the pollution found in soils growing crops that are sold and eaten by the general public. So in order to obtain a soil sample representative of microplastic contamination of the croplands, sediment was collected at each wetland and in the north, west, and south areas of the main floodplain. At each location, a single sample of about 500 milliliters of soil was collected using a shovel. So the top 2 centimeters of the soil was shoveled up and stored into a glass jar and covered with aluminum foil. For the purpose of the study, I chose to look at plastic particles smaller than 1 millimeter. So once the samples were collected, they were dried in a drying oven overnight and then sifted through a 1 millimeter sieve and a 35 micrometer sieve. The soil collected from the 35 micrometer sieve and the soil retained on the 35 micrometer sieve were stored, leaving 12 samples total and 2 samples from each location. Using zinc chloride, I conducted a density separation method to separate the soil from the plastics. The zinc chloride solution was created by mixing zinc chloride and distilled water. It created a solution that was heavier than the plastics, but lighter than the soil, allowing the soil to sink to the bottom as the plastic particles could float to the top. So this is a really helpful diagram to show exactly how I did the density separation. Approximately 5 grams of each soil sample was added to 125 milliliters of zinc chloride solution in a glass beaker. The solution was mixed for about 5 minutes and sat overnight, allowing the sediment to settle and the plastic particles to float to the top. Then the plastic particles were separated by overflowing the beaker with additional zinc chloride, and the overflow was caught in a glass dish and was then filtered using vacuum filtration, and just the plastics were left on the filter paper. The plastic particles on the filter paper were then viewed under a dissecting scope and cataloged into their plastic types, the fragments, beads, and fibres. As discussed on the previous slide, the soil collected from the 35 micrometer sieve and the soil retained on top of the 35 micrometer sieve were stored, leaving the 12 samples total and 2 from each location. The soil retained on the 35 micrometer sieve were considered large particles, and the soil that went through the 35 micrometer sieve were considered small particles. So this is why each wetland and floodplain area have a big and a small section. It allows me to discern between the size of the particles. So after testing, it was found that larger microplastics were in total higher in concentration in each of the wetland sample locations. For example, in the gully sample, there were 18 large beads found, but only 1 small bead found. Similarly, within the west wetland sample, there were 13 large fragments, but only 2 small fragments. This trend was consistent throughout majority of the wetland samples, with the exception of the fibres in the gully and the beads in the east wetland. Similar to the wetlands, the floodplain saw a similar trend where the large plastic particles were higher in concentration, which was consistent within all of the floodplain samples. Additionally, the 3 samples from the floodplains contained more microplastics than the 3 wetland samples. These results partially coincide with the original hypothesis. It was expected or originally thought that small microplastic particles were going to be more prevalent. However, the results show that larger microplastics are found in larger quantities compared to the small particles. So this could be due to the fact that microplastics less than 35 micrometers are more difficult to filter out, or they're just not as common as expected. Additionally, the results show that the beads are the most common pollutant within the Franklin farm, with the exception of the South floodplain. To further the study, a variety of solutions such as sodium chloride, sodium polytungstate, and tap water can be utilized in density separation and may be more helpful to better extract microplastics. Additionally, continuing the testing with soil samples from multiple different agriculture and natural areas can lead to a better understanding of contamination within croplands and help determine whether the data is reasonable. Due to the little research involving microplastic pollution, this project can aid us in understanding microplastic contamination in various areas. Previous studies suggest that microplastic concentrations are higher in areas with higher amounts of water runoff. So by furthering research of microplastic pollution, we may one day be able to apply our knowledge to better understand microplastic pollution and its effects on the environment and how it affects organisms and ecosystems. And that concludes my presentation. Thank you for listening.